Hi, micro folks. We are on our second video for our Bio 440 Lab Chapter 20 protist worksheet. So the first audio we had checked out our free-living um, protozoa, and those included the flagellate euglena, the mixotroph, our ciliated paramecium, and our amoeba proteus. So what we'll do this time around is we're going to look at some symbiotic protozoa, and we'll first start by looking at some parasitic protozoa. So parasitic meaning that um, one symbiotic partner, the parasite, invades the other symbiotic partner, the host, and causes harm. So it's a win-lose relationship. And I thought I would do this according to their lifestyle. So where they live, how people get infected. So we'll first start with, we're first going to start with intestinal pathogens. And we'll look at one amoeba, Entamoeba histolytica, that causes amoebic dysentery. And then under, I'm just looking here, I see I made a mistake. Up here in the flagellates, I forgot to add a really important flagellated intestinal protozoal parasite here in the United States, Giardia. So we're going to look at Giardia. And then for a bonus question, we'll look at, this would be totally for a bonus question, we'll look at a sexually transmitted protozoal pathogen, a flagellate called Trichomonas. So I just need now to find these guys in our worksheet. So let me just back up here. So the Giardia and Entamoeba histolytica will have fecal oral transmission, and then the Trichomonas vaginalis will have sexual transmission. Okay, so let's see if we can't find. Okay, so we're going to start out then with our parasitic amoeba, Entamoeba histolytica. And the specific epithet or species name here, histolytica, literally means tissue lysing. So we'll see that when the entamoeba is living within our intestines, they actually can kill and eat cells of our intestinal mucosa. And so we're going to end up with a bloody, what's called a dysentery. So feces full of mucus and blood. Okay, so let's just do this little guy here. So Entamoeba histolytica. In lab, we're going to be looking at fecal smears. This is one way to diagnose people suffering with amoebic dysentery. And as we'll briefly describe in lab, these intestinal protozoal parasites, they always have two stages. So one stage is the tough resistant cyst stage. This is what we call the infectious stage. It has a really tough resistant cyst cell wall, so when it's shed into the environment, it doesn't dry up. It's resistant to a lot of chemicals, a lot of disinfectants. And it's this form then that if we swallow it in fecal contaminated food or water, the cyst will pass through the stomach. It's not destroyed by the stomach acid, and indeed it's probably activated by the stomach acid. And then when the cyst reaches the intestinal tract, it will exist, releasing the metabolically active, growing, metabolizing, damage-causing trophozoite. So these trophozoites, they're the forms that are actually causing the damage in the intestine. However, they're very delicate. So if the trophozoites are shed into the environment, if they dry up, they're destroyed. Even if we were to swallow a trophozoite, they would be destroyed in the stomach acid. So on our lab exam, if I asked you which form is the infectious form, you would say the cyst form. If I ask you which form is actually causing the damage, you would say the trophozoite. On our lab exam, I'll only show you the trophozoites, right? So the trophozoites are usually the ones that are the most different from one another when we're comparing different intestinal protozoal parasites. Okay, so again, transmission is fecal oral. So again, we think of fecal contaminated food or water, but there could also be literally fecal transmission. For example, maybe in daycare centers where children haven't learned good hygiene after they go to the bathroom, little kids could get feces on their hands, and then they could maybe contaminate toys or put their fingers in the mouths of others. And also when people are being sexually intimate, if there's any kind of fecal transmission, that's another way that these intestinal protozoal pathogens could be transferred. So in the United States, there's a relatively low occurrence of Entamoeba histolytica. When they do antibody surveys in the United States, less than 1% of folks have antibodies against Entamoeba histolytica. It's not endemic here in the United States, but if you have folks that travel around the world, maybe visiting countries where there isn't maybe clean sources of drinking water, that might put people at risk for becoming infected with Entamoeba histolytica. So in some areas of the world where there aren't good water treatment um, um, facilities, there can be infections rate of 5 to 10%. So out of every 100 people, 5 to 10% of them will be infected with Entamoeba histolytica. And another challenge is that you can have asymptomatic carriers, so people can be shedding the cysts in their feces and not be showing any clinical signs themselves. So just some tidbits here, folks. So the trophozoites of Entamoeba histolytica, they like to colonize the colon, and there they're going to attach to the surface of the mucosal epithelial cells, and they insert these proteins into the cell membrane of our intestinal cells. And it's so cool, they're called amoeba pores. So they form pores in our intestinal cells, and that causes our cells to die, to lyse. And then the amoeba eat our cells, right? Isn't that amazing? So this will result again in a bloody dysentery, so feces with blood and mucus in it. Some folks describe the feces as like raspberry jam feces, and I'll try to have a photo of that in lab. Because the entamoeba are actually destroying our intestinal cells, they can spread to other parts of our body. So one concern would be if they spread to the liver, they can form liver abscesses, right? So these guys can spread, so that makes them more virulent than when we talk about our Giardia that's really common here in the United States. So just a couple of notes here, you guys. So in people that aren't immune, there's going to be a white cell response, a leukocyte response, and the amoeba can kill and eat our neutrophils and macrophages if the macrophages haven't been activated. So in people that are immune, it's a cell-mediated immune response. They activate their macrophages to turn them into what we call angry killer macrophages. So there can be a CMI response. You want to note that if the patient is on steroids, that's going to reduce the cell-mediated immune response. And let me see, what other little tidbits do I have here? The amoeba also make an enzyme called a cysteine protease, and that means they can hydrolyze, they can digest the secretory IgA and IgG. Secretory IgA is one of the main antibodies that are involved in mucosal immunity. With regard to diagnosis, you can take a fresh fecal sample and look for the trophozoites or the cysts in the fecal sample. You could do a scraping of the colonic ulcers via an endoscope and again look for the trophozoites. In addition, there's a immunoassay where you take feces from the patient and you run your immunoassay, which involves antibodies to the Entamoeba histolytica antigen. So there's an immunoassay available. And also you can check the serology for antibodies. And again, in the United States, less than 1% of folks have antibodies against Entamoeba histolytica. So if you have a patient with a really high antibody titer against Entamoeba histolytica, that might suggest that they are indeed infected. All right. So now let's see if we can find our Giardia, which is the second intestinal. Here we go. Here's our Giardia. And again, folks, I apologize. I forgot to put this on the table on the front page. But folks, Giardia is really important here in the United States. It's the most frequently diagnosed parasitic intestinal disease in the United States. We definitely have it here in California. And again, people at high risk would be maybe backpackers or hikers that are drinking water from streams or lakes. We have to just presume, folks, that all of our fresh water here in California has probably been contaminated with Giardia cysts. Also, children in daycare centers or people that work in daycare centers are going to be at high risk. And again, anytime where we have intimate relationship where there could be transfer of feces into the mouth, that would also be of huge concern. Apologies, our cat is meowing in the background. So it's a zoo here, you guys. All right. So we're going to be looking at fecal smears. And again, folks, on our lab exam, I'll only have examples of the trophozoites. So just like with Entamoeba histolytica, Giardia has two stages. It has a tough resistant cyst stage. This is the infectious stage. And then once we swallow the cyst, it's activated by stomach acid, and then it excysts and releases the metabolically active, growing, dividing trophozoite in the intestine. So the cyst is the infectious stage. The trophozoites are the ones that are going to cause damage in our intestine. So this is a stained example of a Giardia. And you can see it has what I consider like a teardrop shape here. What's really neat about them, if you see them under the microscope, if they're oriented correctly, they have two nuclei. And these almost look like little eyes that are staring up at you, almost like a Gary Larson cartoon. They have 4 paraflagella. We can just barely see them here, so they are flagellates. And the way they colonize our intestine, and again, these little guys are going to be going for the duodenum. They have a ventral sucking disc, and so they use that to attach to the surface of our intestinal epithelial cells. And what can happen is if there's large numbers of them, they can pave the surface of the duodenal mucosa. And as a result, we'll end up with malabsorption conditions, because nutrients aren't being absorbed. The feces can be foul smelling, a fatty feces. And one of the books said with a fatty feces, it will float. Like, so if your patient defecates into the toilet bowl, the feces will float from the high fat content because the fats aren't being absorbed. Excuse me, in chronic infections, the patient can lose weight. So again, you can have this wasting syndrome. Because Giardia lives so high up in the intestinal tract, if you take a fecal sample, you might not always see the trophozoites or the cysts. And indeed, they say if you're going to be using microscopic exam of the feces to try to diagnose Giardia, you have to take at least 3 different fecal samples at different times because the Giardia aren't always shed constantly. In addition, again, there's an immunological assay, an ELISA assay, that you can run on the feces to detect Giardia antigens, and that's supposed to be about 90% sensitive for that. This is really disturbing, you guys. The cysts are so resistant that normal chlorine levels that are used in water treatment facilities aren't sufficient to kill the cysts. So there's actually been outbreaks of giardiasis that are associated with properly treated water, quote unquote. So that's kind of disturbing. One of the therapies that's used to treat Giardia, and I've seen 2 discussions on this drug, metronidazole. So metronidazole, it's a drug that's often used to treat anaerobic bacteria such as Bacteroides. So I was reading up on why this is. So in a nutshell, metronidazole, when we give it to our patients, it's in an inactive form. And for it to become active, it has to be partially reduced. And so anaerobic bacteria are really good at doing this partial reduction. And I'm thinking it's because the metronidazole, it's going to act as an alternate terminal electron acceptor in anaerobic respiration. So we get partial reduction of the metronidazole to the active form. And in the active form, it becomes incorporated into the DNA of the pathogen. So this could be the anaerobic bacteria, or in this case, our anaerobic Giardia. The intestinal tract is anaerobic, so the Giardia, just like Entamoeba, they have to be able to grow anaerobically. So when the activated metronidazole is incorporated into the DNA of the parasite, the DNA becomes unstable. And this is referred to in one of the microbooks is referred to as lethal synthesis. So as the DNA is unstable, then this is going to kill the parasite. The only trouble is that sometimes metronidazole can be activated in our tissues, even though we're carrying out aerobic respiration. You can get some activation of metronidazole in the human tissues, and this is of concern because if it then gets incorporated into our DNA, there's a concern that it could increase mutagenesis and maybe um, um, be considered a carcinogen. So I'd read in one book where, and I'll double check this. One book was saying, yeah, metronidazole is a drug of choice, and then another book was saying it might not be available in the United States. So I'll try to do some more research on that one. Okay, so metronidazole would be possibly one of the drugs to treat the Giardia infections. There can be relapses if you're using metronidazole, and my understanding is it might not reach high enough levels within the intestinal lumen. So there are other drugs such as tinidazole and nitazoxanide that can be used if you have relapses, and I'll try to find out, maybe those are a little bit safer medications. The metronidazole can also be used to treat entamoeba histolytica infections, and it's said the metronidazole is well absorbed into tissues, so it's really good at killing the invasive entamoeba histolytica, but again, might not get really therapeutic levels within the intestinal lumen. Okie dokie. So that's our second intestinal protozoal parasite. This is just showing this cyst. Again, this fecal smear that's been stained. This is the tough stage, the intestinal phage. Giardia, a lot of people consider it a zoonosis. One of the former students was from Canada, and one of his jobs had been to leave canoe expeditions. And he said, yeah, sometimes he and his clients would contract giardiasis on these canoe expeditions, and he said they called it beaver fever. And one reason for that is some people believe that beavers are a reservoir of giardia. Often wild animals get blamed for being the source of the giardia, but we want to remember that if you're hiking, often people, if they need to defecate, humans will defecate improperly. So if you're going to defecate out in the wild, you need to dig a deep hole and defecate into the hole and then cover it up. So if humans are defecating like on the surface of the ground, then if it rains, if there's any giardial cyst, that can run into the creeks and rivers and lakes. Or if they're just making a very shallow kind of latrine hole, again, if the feces isn't very deep, if there's rainstorms or if there's snow and then snow melt, then those cysts from the feces again can get into the streams, the lakes, and our waterways. So probably a lot of the outbreaks, it's not wild animals' faults, it's probably humans' fault. All right, folks. So that's our little Giardia. And then we'll save the arthropod-borne, the blood parasites, folks, for the next video. So we'll talk about the trypanosomes and plasmodium. And let me see. I just want to see here if I got Trichomonas in here. Looks like probably I didn't. Okay, so folks, just really briefly as a bonus question, there is one sexually transmitted intestinal, no, sorry, not intestinal, apologies, you guys. There's one sexually transmitted protozoal parasite. This would be a bonus on our lab exam. And this parasite is called Trichomonas vaginalis. It's the most commonly diagnosed protozoal urogenital tract infection in humans. And this might be worldwide, but certainly here in the United States. So we'll have some prepared slides, demo slides, you guys, of Trichomonas vaginalis. It's a pear-shaped flagellate, and it also has a cool undulating membrane. In females and women, it causes inflammation of the vaginal, vulvar, and cervical mucosa, and you can get this copious yellowish malodorous discharge, meaning foul smelling. Less frequently in males, you might get infection of the urethra, the prostate, or the seminal vesicles. And in men, instead of this copious yellowish malodorous discharge, there might be a white discharge. It's, as we mentioned, usually it's sexually transmitted. And if one partner is infected, then all partners should be treated. Of interest, and again, this is homework for me, folks. Um, the medical microbook was describing that normally in the female vagina, the pH is around pH 4. And this is probably due to all of our good lactic acid bacteria that colonize the vaginal mucosa. And it turns out that Trichomonas vaginalis doesn't grow very well at that low pH. It grows best at around pH 6. So the microbook was saying that if a woman has an abnormally alkaline pH of the vaginal mucosa, and I was trying to think how would that happen. Well, maybe if she's been on antibiotics and it's killed all the good beneficial lactic acid bacteria, that might permit the vaginal mucosa pH to increase. That would increase her risk for becoming infected with Trichomonas vaginalis. One way to diagnose it is to take secretions from the urethra or from the vagina, make a wet mount, and apparently these little guys will be motile. I've never seen them live. So you can diagnose just by making a wet mount. You can also do a stain, which that's what we'll have as our demoscopes. And they said you can also culture these little guys, which I thought was kind of interesting. Again, one of the recommended drugs was metronidazole, which we had described previously in talking about Giardia and also entamoeba histolytica infections. So folks, I think that does it for this audio. So this was our 2 intestinal protozoal pathogens, entamoeba histolytica causing amoebic dysentery, Giardia, which causes giardiasis. Giardia, folks, does not cause a bloody diarrhea. They don't invade, they don't spread, they just live in the intestinal tract. So it's just fatty, foul-smelling diarrhea with maybe bloating and abdominal discomfort. And then we'd finish with a quick overview of the urogenital protozoal pathogen Trichomonas vaginalis. Okay, we'll do another video for the arthropod-borne, the blood-borne pathogens.